Hello. Yep, as I said, I'm Andrew Walter. I'm going to be talking about some work that's kind of spun out of some stuff happening at York and Ulster, where we've been looking at artificial neural microcircuits. So first of all, hang on. Come on. Please move. Thank you. So first of all, a small bit of context. This work has come under the auspices of something called the Nervous Project, where we're looking at using spiking neural networks as fault amelioration systems for systems on chip. The sort of idea is to kind of give systems on chip a kind of artificial nervous system, where the idea is that will pick up the sort of pattern, pick up the sort of signs of a fault about to occur, and then take ameliorative action. So we were digging into more specific biological inspiration for this concept. We came across discussions of this idea that neuroscientists term neural microcircuits, which is in various model organisms. In particular, we have a American crayfish, which is one of the ones they like to use. There's identified the fact that throughout their peripheral nervous systems in particular, there are certain repeating subunits or patterns of neurons that perform particular functions. So, for example, here we've got the anterior section of the tail, which contains a lot of the nervous system that's responsible for locomotion in the crayfish, and it contains repeating versions of these two microcircuits. On the top, there's the microcircuit that controls sort of standard locomotive behavior. You have this microcircuit sort of repeated down the animal on either side. They generate sort of complex patterns that power the propulsive limbs, which referred to as swimmerets. But then below it, you've got an example of what's called the fast escape or the tail flip escape reflex microcircuit. And again, this microcircuit is repeated down the animal either side, and it responds to a quite complex series of sensory stimuli to determine whether or not the animal is under threat. And if it is, it then triggers a large reactive muscle movement. So obviously we were studying these with the intent of sort of, okay, we want to do something similar. We want to have small elements that we can insert into chips to do particular tasks. But it then sort of calls us to ask, well, in animals, these microcircuits then assemble together to form the larger gross peripheral nervous system, which, as you saw, can do quite complicated things on its own volition. So could you take that same idea of building a catalog of artificial microcircuits that perform particular functions that you can then build together to form a larger, more complete neuromorphic system? So we started sort of looking into this idea. So the first part of this is what we need to do that. What do we need to assemble this hypothetical catalog? So we would need, obviously, building blocks for these neural microcircuits. We need something to use as our fundamental unit. We need some way of saying, well, what does microcircuits look like? How do we build them? So you can then, take that and use that as a blueprint to put the microcircuit wherever you want. And then we need to actually start looking for useful microcircuits, because sitting down and sort of manually trying to design this sort of thing is a very intractable task, particularly when it comes to in our case, specifically with spiking neurons. Spiking neural networks can be a bit finicky, a bit unusual. So starting with the first of those questions, the building blocks. Now, one of the things we wanted with this idea is that we want to be substrate agnostic. So in other words, we didn't want to say, these work if you use this neuron model, especially because with spiking neurons, you can get quite a broad variety of different behaviors from specific neuronal models. So instead, we sort of went one step up in the biological world and looked at these things called neural motifs, which sort of neuroscientists identify as kind of common recurring patterns you'll see in the nervous system. So we've got a few of them here, the sort of very simplistic ones. We've got feed-forward excitation, obviously a signal, a spike train enters and is fed forward. Feedback excitation, where a spike train will sort of curve back on itself and provide additional stimulus to keep it going. Feedback inhibition, that one's, again, the idea this spike train feeds back on itself and sort of dampens further behavior. We've then got these recurrent elements, sort of cyclical behaviors, both excitatory and inhibitory. And then we've got a couple of sort of unusual ones, a couple of wild cards that we used. Lateral inhibition, which is the idea of you have sort of two strands of signal in parallel. One of the strands can then inhibit its neighbor if it gets there. And the other is a sort of classic recurring nervous system elements is a central pattern generator, which is effectively a sort of neuron oscillator. It generates a sort of consistent signal. So with those as our starting point, we drew up these little connection matrices to describe each of these. As you can see, for example, on the top with the central pattern generator, you've got neuron 1 is connected to neurons 1 and 2, and neuron 2 is connected to 1 and 3, and so on and so on. And with that, you can then just sort of staple these connection matrices together and build up a hypothetical microcircuit. You will note in the connection matrix, the example connection matrix on the right, that there's obviously the one labeled as W's. Those are hypothetical weights those are hypothetical connections you could have to link the two microcircuits together. But also we've got these inepitables, these dummied out ones, which are the case of where that connection would not be allowable. So, for example, because we have defined explicit input and outputs on the motifs, we wouldn't want, say, a connection that was trying to go into an output or come out of an input. So that just covers that. And together, you can see you sort of take the connection matrix, like the one on the same on the previous slide, and add a few other things to just describe your sort of input and output connections of the whole thing. And there you have a hypothetical one of these artificial neural microcircuits. So that's all well and good, but what does it do? Is that useful in any way? So we therefore want to sort of assess the behavior of these microcircuits, and from that then determine, is this something useful? Is this something we put into our hypothetical catalog? So the approach we've taken so far for sort of this preliminary experiments is we generated a spiking 8-bit bus, for want of a better term, just because it was easy. We could use a string of text, we could encode it, we could transform it into an 8-bit encoding, and then use that just to generate bursts of spikes. So you can sort of see how that propagates through time. We then feed that into, the inputs of a microcircuit. We went fishing deliberately. We said, okay, let's go look at eight input microcircuits and see what we get. We then obviously get some sort of output. We get the spike trains that come out of that microcircuit, and we then want to do something with it. The first question is, can we use that to sort of put a label on the behavior of the microcircuit? So in much the same way that we have plenty of electronic components in existence nowadays, where you can look at the data sheet and say, oh yes, this transistor has this behavior, so I know what circuit I want to use it in. We want to say, can we put a label on a microcircuit and say, oh, it's a 4B microcircuit or something like that? And the other thing is, obviously, even before you get to that stage, we want to be able to answer that question of, is this a useful microcircuit? Does it do something new, worthwhile adding to the catalog? So it turns out that first possibility is actually quite difficult, and we hadn't actually figured out a method to put that sort of nice, convenient label onto a microcircuit, onto the behaviors yet. But that second one, the idea of differentiating between different microcircuits, it turns out neuroscience has a whole grab bag of techniques for doing exactly that, in the form of what they call spike distance measurements, which are basically, you can take two spike trains, you can feed them into one of various different algorithms or different approaches, and you'll get a value that indicates how dissimilar the two spike trains are. The one we decided to use when we gave this a go is something called bivariate spike distance, simply because it's quite nice for this purposes. It spits out a number between 0 and 1, and it will only ever be 0 if the two spike trains are completely identical. So if you want to compare, our two microcircuits, we want to say how different are they, that's quite a good metric. The exact mathematics there of bivariate spike distance, it is a bit Byzantine. The long and short of it is, you sort of chop your spike train up into sample intervals. At each interval, you take a number of measurements related to the next spike and the previous spike immediately before. You use this to calculate an array of what it calls instantaneous spike differences, which are effectively a sort of a difference at that moment in time between your two spike trains, and then you integrate over time to get your singular value. So we have those nice convenient single values. We can then use those, it turns out nice and easily, to automatically go fishing for useful microcircuits. And we were doing this with novelty search. So novelty search is a bit like sort of standard evolutionary or genetic algorithms, but rather than defining some sort of fitness function, rather than saying we want it to be like this, you say, just show me things I haven't seen before. And as you can very readily imagine, with a big pile of those spike distance measurements, it's very easy to ask the question, is this microcircuit different from other microcircuits? You can just basically take a microcircuit, calculate differences, for the other microcircuits you've already seen, average that out, and that's pre-packaged effectively a nice novelty value, which then sort of all stapled together gives you this kind of a workflow. You randomly initialize a big pile of possible microcircuits. You feed them into some sort of simulator. In our case, we use Brian because it's easy and straightforward to use, strictly Brian 2. We feed in some sort of stimulus, again, in this case, as before, that those 8 spike trains acting a little bit like an 8-bit bus. You get the output spike trains, you run them through the Bivariate spike distance calculator and get this nice big matrix that compares all your microcircuits with all the other microcircuits. You get your you compact that down to your single novelty value, and if it passes the threshold, whatever that may be, that's a microcircuit. Let's put it in our catalog and maybe use it to do something. So we did a run of this. We executed a run of this algorithm. Population of so obviously it's a process. We had a population of 150 generations, so it's effectively assessing 5,000 potential microcircuits made up of those motifs that I showed earlier, being fed that stimulus that we showed earlier, and of those, 50 got moved into the catalog as being declared novel. So from some basic analysis, although you could then sort of break these microcircuits down to 4 categories, we had a few microcircuits that were effectively constantly generating some form of spikes. Not obviously, consistent solid spiking, because then they wouldn't be declared different enough from each other, but various forms of oscillatory behavior. And broadly speaking, that's strongly correlated with the presence of central pattern generators in those microcircuits, which seems reasonably an obvious thing to note. We then had what we called weaking spiking microcircuits, which are effectively microcircuits that generated output spikes on some instances of a particular pattern of input stimuli, but not all. So, for example, the one in the middle, because we were using a text string to initially generate those 8-bit patterns that became the input spike trains, we could sort of draw this nice, clear to explain correlation. So this is a microcircuit that spikes on some of input pattern P, but not all the examples of input pattern P. So not necessarily the most useful thing, but that's what it had. We then got the 7 strong spiking microcircuits. Those are similar to the weak spikings, but they spike consistently on one or more of the input patterns, which is that shows a good sort of fact that these microcircuits they generate a clear correlatory behavior. And then we had a few microcircuits that exhibited some mixture of strong and weak, so they might strongly spike on one input pattern, weakly spike on another. So if we then sort of look at some of those strong spiking microcircuits, which are, as we said, those are the ones that seem most immediately useful, we got 2 very nice ones. We got microcircuit 392, which is this one here, which responded strongly to inputs that look like a dash, so the dash character equivalent. And we got microcircuit 466, which strongly responded to the various punctuation characters. It sort of latched onto those as a group and responding to all the characters in that group. And you can sort of, I think, see how components like that, things like that, could then be used to build up larger neuromorphic, sub-elements of larger neuromorphic systems. Obviously, what we've presented here today is effectively a proof of concept, initial tentative thing. Further work is needed to sort of fully develop this idea. We are working on this. Most particularly, we're working on improvements to the generator algorithm that we showed, in that we've now got a broader handle on the sort of the motifs that neuroscientists talk about. There's turns out they're a bit more of a broader church than the ones we've got. And also we're making some discoveries about the importance of exactly how you sort of choose your stimulus for testing your microcircuits, which we are getting into. And of course, we still have some open questions. That open one about, is there a convenient way we can label the behavior of microcircuits in a very sort of explainable fashion, which we've still not found a satisfactory answer to? And also kind of as a connection to that, is there a way we can trivially visualize the behavior space of the microcircuits we've got? Because obviously if you try and sort of decompose these spike trains into some sort of X number of dimensions, you'll get quite a high dimensionality, which isn't easily plottable. But is there some other metrics we could use to sort of plot down this catalog of microcircuits and say, okay, there's some down here, and so on. But yes, that is the state of things there. That is all that. Thank you very much.